It's a great pleasure to introduce today's speaker is Bertrand Delgutte. Bertrand is somebody that I've known virtually all my career. We've worked on similar problems most of this time, the main difference being that Bertrand's done a better job at solving them. Bertrand got his degree at École Polytechnique in Paris and came to MIT for his PhD and worked there with Nelson Kang and the other people in the Peabody lab, and he's pretty much been there for the rest of his career except for 3 years that he spent working for the French telephone company. And as you can see, he's got appointments at practically every institution in Boston, so I won't try to list them. And what he's going to talk about today is the representation of complex signals in the midbrain auditory system.
Well, thanks. I want to thank Fred Jelinek for bringing me here. I've certainly enjoyed my day. It's fun to talking to people. And I want to thank Eric Young for babysitting me all day. So I was telling the students earlier that when I came to MIT as a graduate student in the mid-70s, I came with the intention of working on speech recognition. And I quickly let's say, get discouraged with it. I was afraid I would have to tinker with parameters too much. But they convinced me today that I was wrong, that it was only my ignorance, that since I didn't know the hidden Markov models, that solves all those tinkering problems. So as a result of that, I switched to another field, and I learned to do auditory physiology. And I did my thesis on how the auditory nerve responds to speech. And as my career went on, my interest broadened, and you see now in the title is music. And it's not only the auditory nerve, so most of what I will be talking about today is actually what goes on in the brain, in the parts of the brain that respond to sound. So I have a very ambitious outline in two parts. First, I will talk about the coding of the temporal envelope of speech, and I will define that in a minute. And looking at how that temporal envelope, which I argue is very important, is represented in auditory nerve fibers, which are the output of the ear and the input to the brain, and also how it is represented in auditory neurons in the brainstem and the midbrain. And I will see if we can come up with a model that predicts the coding of the temporal envelope in speech. And then I will switch gear and talk about another aspect of the temporal envelope, the role it plays in music. And I would say that the temporal envelope is the basis for the transition of roughness, which are one important contribution to the musical consonance, actually the dissonance of musical sounds. And I will show that there are clear differences in the auditory nerve and midbrain auditory neurons as to how consonant and dissonant sounds are represented. Okay. So I say what ties the two parts together is the temporal envelope, that aspect of the stimulus which I call the temporal envelope. So what do we mean by temporal envelope? So this is this sort of defined in a simple way. You can take an arbitrary signal. This is here a bandpass filtered excerpt of speech. And you can divide it as a product of two signals, an envelope signal, shown here, and a fine structure signal, so the envelope signal you can also call the modulation or modulator, and the carriers, the fine structure signal, you can call it the carrier. So this is simple. The question arises when we wonder what signal should we put here as the input. So one possibility would be to just take the acoustic stimulus, the sound pressure at the eardrum. In fact, if you do that, you find that temporal envelope is actually exceedingly unimportant. And that was shown many years ago, in the 40s, I think, by Miller and Licklider. we did some experiments where they took speech and they did infinite peak clipping. So instead of having something with a nice envelope, everything was compressed. So you had something infinite peak clip, and they found, much to everybody's surprise, that infinite peak clip sounds terrible, but it's intelligible. You can understand what it's saying. So this operation destroys the broadband temporal envelope, but it still preserves intelligibility. So if that were the only story to the envelope, there would not be much to it. Fortunately, we can redefine it in a way that makes more sense, because the nervous system, the auditory nerve, never sees the actual acoustic waveform. It sees the waveform processed through the cochlea, and the cochlea works like a bandpass filter. So essentially, each place along the cochlea will see a bandpass filtered version of the stimulus. And what's important is that the action of filtering can transform fine structure information into envelope information. And that's shown by this slide. Oops. Unseparated slide. So we start from the top panel shows the raw unfiltered stimulus, and that's a frequency chirp, right? So over 40 milliseconds, the frequency changes from 1 to 2 kilohertz. So this stimulus obviously has no envelope information. It's all fine structure. Now, if we put that through two different filters, so one filter is centered at 1.3 kilohertz and 1.7 kilohertz, so this could represent the response of the basilar membrane at two places on the cochlea. You see that the very action of filtering causes now some envelope, the envelope to become maximum at some point in time. And if you had a bank of filters tuned to all the frequencies between 1 and 2 kilohertz, you could perfectly, by looking only at envelope information, you could reconstitute the frequency trajectories of the chirp. So once we filter the signal, envelope information is actually becomes important, and it actually and that's illustrated partly in the next slide, which is thanks. Okay, so this we now go back to real speech. So these are three ways of representing speech. So the first, this is an utterance, wood is spent for making toys and blocks, and the top is the well-known sound spectrogram. The second trace is the broadband, broadband temporal envelope, or on this time scale, we could also call it a waveform, which I argue is not essential for speech intelligibility. The third panel shows an envelope power spectrum, and to do that, I split the signal into a large number of frequency bands to sort of mimic the filter bank action of the cochlea, and for each filter output, I computed the temporal envelope, and then I did the power spectrum of this temporal envelope, and then I summed the temporal envelope for all the different filters. And what you get when you do that is this. You get basically a low-pass spectrum, and the frequencies, there's a peak at 3 to 4 kilohertz, and that corresponds to the average rates between syllables. So basically, the syllables is a vowel and a consonant. The vowel is louder than the consonant, so you get pronounced amplitude modulation here at 3 to 4 hertz. And so after this peak at 3 to 4, the spectrum falls gently, and you see another maximum in near 100 hertz, which would be perhaps appropriate for the pitch of voice for a male speaker, which this was a male speaker. So people have tried to wonder which part of the spectrum are important, and what aspect of the temporal envelope exactly defined this way is important. And they found that it's a low frequency in the envelope that are the most important. Frequencies below roughly 20 hertz are the most important for speech reception. So this shown here. So as I just showed, speech show major changes in amplitude envelope that result, well, pronounced envelope modulation at low frequencies, as shown by this modulation power spectrum. And some experiments in which speech is degraded by either background noise or room acoustics show that degradation in low frequency information correlates with degradation in speech reception. On the other hand, Bob Shannon and his colleagues a few years ago did some experiments that show that speech reception is possible with very degraded spectral information, as few as 4 frequency bands, so long that the temporal envelope is preserved in each band. And also, we know that auditory neurons in the central nervous system are selective to particular envelope frequencies. So this suggests that it's well worthwhile to study how temporal envelope is coded in the auditory nervous system. Okay. So I will go back to the slides. So I will get into the main story now, how we do those experiments. So these experiments are done by recording from single neurons in anesthetized cats, and this shows the brain of the cat, and the cerebellum has been removed as part of the occipital cortex to expose the auditory brainstem here. And it turns out, it so happens that a lot of the auditory structures in the brain actually lie on the dorsal surface of the brainstem, so we can see it on this picture. So all the information arrives here by the 8th nerve, or auditory nerve, which represents the output of the cochlea. So part of my recordings will be done here, since it is the output of the ear and the input to the brain. All the information about the acoustic world, everything that you can hear, has to be encoded in the auditory nerve in some way. The next step that I'm going to show, actually a limited amount of data, is the cochlear nucleus, which is sort of the first center or the center in the brain. And what's interesting about the cochlear nucleus is that information is split into different channels. You have different types of cells which respond selectively to different aspects of the stimuli, and each one is part of a different pathway that transform the information to the higher center in different ways. So I will show some data from this also. The third place I will show data is this inferior claviculus, which is the principal auditory nucleus in the midbrain. And that's after this sort of specialized processing starting in the cochlear nucleus, everything comes together into one big nucleus. And then the information is again transmitted to ultimately reach the auditory cortex, which in cats is on the surface, in humans it's very deep into a fissure. Okay, so I will be showing coming from the auditory nerve, the cochlear nucleus, and the inferior. I will skip this one. So this is the only slide that shows about what goes on in the ear. So it's my sketch view of how the ear works. So the snail-shaped thing, sorry, it's in French, figure it out. So snail-shaped symbol representing the cochlea, right? The auditory part of the inner ear. This thing, this trace is shown recurring from different neurons in the auditory nerve to one stimulus, and the stimulus is shown on top. So it's a stimulus, if you want, that has some envelope and some carrier information. So this stimulus resembles a vowel, it's a very simplified vowel. And the neurons are arranged in a way that neurons that innervate the so-called base of the cochlea. So those neurons respond to high frequencies and innervate the base of the cochlea, while the neurons at the bottom respond to low frequency and innervate what's called the apex, the other end of the cochlea. And as you know, the cochlea does a frequency to place mapping such that each place in the cochlea selectively responds to a range of frequency. So when we look at this, we see that the auditory nerve, most auditory nerve fibers that are shown here, once they respond to the stimulus, they actually encode both the fine structure and the envelope. You can suddenly see that here, peaks at one time, just like this did here. This neuron also responds only to the solid envelope onset, while some of these neurons here show fine structure information as well as envelope information. So in the auditory nerve, at least for frequencies in the speech and music branch, both fine structure and envelope are represented. Now when you assign the auditory system, the fine structure information is rapidly degrading. And by the time you reach the auditory mid-grade inferior colliculus, you find few neurons that respond to the fine structure. So that's why we focus the rest of the talk on the envelope information. So how did we study that? So from the same neurons, whether they were in the auditory nerve, the cochlear nucleus, or the inferior colliculus, we did two sets of measurements. One measurement was simply to obtain the response to a speech utterance. And the second measurement was to measure what we call a modulation transfer function. And the modulation transfer function is meant to characterize the envelope processing performed by the neuron. And this slide shows how we measure the modulation transfer function. So the stimulus is shown here. We use a noise which is modulating, sinusoidally modulating, as shown. So the red represents the actual noise and the sine wave top sample. We measure the neural response of one neuron to this, and that's what's shown in blue. And then we feed the sine wave to the neural response in Fourier analysis. And then we look at two things. First, what's the size of the sine wave relative size, normalized size relative to the sine wave in the stimulus? And that gives you, for one modulation frequency, that gives you the magnitude of the modulation transfer function. And we also look at the delay. You note that the maximum here in the response is going to occur at the same time as in the stimulus, so we get the phase shift or delay of the sine wave in the neural response relative to the stimulus, and that gives us 1 point of the phase versus modulation frequency. So for each modulation frequency, which means that we obtain the magnitude point or the phase point, and then we repeat the same measurement for a whole bunch of modulation frequencies, ranging from 1 Hertz up to 500 Hertz. And then we obtain the symbols here, shown in the 99 phase, and then we fit the filter model, a lower order filter model to the data. And then we invert the time. No, we inversely transform the 99 phase of the filter model, and then we integrate in time to obtain what we call the step response. And the step response, if the system were linear, represents the expected response to a step in intensity, an abrupt increasing intensity of the stimulus. And in this case, the step response corresponds crudely to the response to the step in the intensity step in the noise curve coming from the same neuron. So how do those modulation transfer function compare in the, well, first what do we see? What kind of modulation transfer function do we see? So So these are data from 2 neurons in the inferior colliculus. So one neuron on top and another neuron at the bottom. And we see on the left the magnitude of the modulation transfer function, the phase of the transfer function, and the step response, like derived from the Richter model. And the 2 neurons have very similar magnitude functions. They both what we call band pass, and they respond preferentially to modulation frequency near 50 Hertz, right? In its broad range of modulation near 50 Hertz. And on both sides of that maximum form. So the magnitudes resemble each other. The phases response are dominated by a straight line, which is a pure delay, which is just the time it takes for the spikes to arrive at this neurons for the conduction time. And the step responses are very different. One neuron, that's what I call the monophasic step response, which resembles the response to a step in intensity as you would see in the auditory nerve. And here you see a transient response at the onset followed by a decay or adaptation. So this is coming from the inferior colliculus, but this cell or step response is what you would see and what you always in fact see in the auditory nerve. This other neuron, even though it has a similar magnitude, the step response is very different. You have a transient positive excitation followed by positive phase followed by a long negative phase. So one possible interpretation of that is you have to have a brief excitation followed by sustained inhibition, and such things have been actually seen when doing intracellular recordings in the inferior colliculus. So this is what we call a biphasic neuron, which means you have a positive phase followed by a negative. So because the magnitudes are similar, the step responses are different, there must also be differences in the phase. And if you look closely, although it's hard to see, most of the difference is actually the initial phase. Here there's an initial phase of pi, and there is an initial phase of 0.
Bertrand?
Yes.
This is a linear analysis.
Yes.
And so if you were to predict the history thing on the right.
Yes. This is what you expect to be the PSC story.
And the notion that it's inhibitory means
you would not see, but when you modulate, right, you have some sort of step, you have some sort of sustained activity, so you modulate. So actually I think this may be a way to reveal inhibitory activity. I mean, I'm not saying it is inhibition, I'm saying it's one way to think about it. I have no way to say what is negative phase. It's just a result of my system analysis, right? But now the simplest explanation I can give it is that it's inhibition.
But your system analysis is linear.
So why do you say that inhibition is non-linear?
No, all I'm saying is that if your arguments are correct, then inhibition should be linear.
I'm not saying that anything that...
I'm not saying it's not.
Okay.
That it's not.
We'll see whether it's linear or not later, right? That's one of the points I think you make. Okay. So let's reserve the judgment on whether it's justified to do linear analysis. Let's just see if it works. Okay. For the moment, this is what I see with this particular sinusoidal modulation. Yes.
So when you calculate this transfer function, is it from the input to the cell or to the neuron?
So it's, no, we can't do that. Yeah, that would be very nice to do that. So it really is a modulation function of the entire system, from the eardrum to the neuron you're recording from. You know the sound you put at the eardrum, and you record from a neuron far away in the system. It could be a dozen neurons between, or maybe not that many, but half a dozen certainly. And it's the modulation function of the whole system. Yeah, that's a very, very important point. Yes. Yes, no, actually it was the speech shaped noise. So my goal was to predict response to speech, so I used a noise with a speech shaped spectrum, average long-term average spectrum of speech. Low pass, if you will. much all the neurons respond to that stimulus. There may be a couple of exceptions. Yeah. Well, maybe a very high frequency neuron might not respond, obviously, because it's low pass. But the neurons in the speech frequency range is crazy. Yeah, noise is, I found, a very effective stimulus. Yes. So we did this analysis, right? This MTF measurement for a whole bunch of neurons from the auditory nerve, the cochlear nucleus, and inferior auditory. And here just shows what the typical neuron is. Right? It's we feed it parameters to all of the neurons in our cell, and these are sort of models that have the median values of the median model parameter values. And so green is the auditory nerve, red the cochlear nucleus, and blue is the inferior nucleus. And you see that all three functions are bandpass, right? They all prefer some range of modulation frequency. You also see that the range of modulation frequency that the neurons prefer decreases from the auditory nerve to the cochlear nucleus to the inferior nucleus. And you also see that the modulation gain, the amplification of the modulation, increases as you ascend. So as you ascend, the neurons are more and more interested in modulating stimuli relative to what goes on in the periphery. Yes?
You find any small sound pressure level?
We have in the auditory nerve, we looked at it, and we do find some. We haven't done it in the other places.
Which is the auditory nerve?
But it doesn't shine what?
I just can't see the color.
The green, sorry.
It's my eyes, it's not your
This is the auditory nerve here.
You get better high frequency modulation higher up the system.
No, low frequency. When the gain is less, if you normalize the gain, this would have a higher curve. The green and red, these two curves have the same gain. This one would have some higher frequency. But that's not, this part is not too interesting. It's just lots of synchrony. So it's not. But what's interesting is what goes on at low frequency. I guess it's most important for speech. Okay, so and here we see the step response of the sort of average spectrum of the auditory nerve, the cochlear nucleus, and the inferior nucleus, and we see that whereas the typical auditory nerve and cochlear nucleus neuron are monophasic, they usually only one positive phase. Okay, the typical IC neuron is biphasic. So this is all we say about the modulation transfer function. Now I will move and look at responses to speech, and then I will try to put the two together. Oops. Thanks. I think this is a little dot.
Yeah, there's a little dot.
I added that one at the last time. Okay. So this is actually another reference. Joe took father's ring Chubin-Charles, and here we see the spectrogram. And the stage tries to visualize the response of the entire auditory nerve to this utterance. So basically, each trace represents the response of the one auditory nerve fiber, or an average of the responses of several auditory nerve fibers with similar bass frequencies. And the auditory nerve fibers are arranged from low bass frequencies. This would be the fibers innervating the apex of the cochlea, or on top you see fibers innervating on the base of the cochlea. And basically, the point of this figure is that the onset transients, which are shown by arrows in the spectrogram, when you get an abrupt increase in intensity in either at low frequencies in red or at high frequencies in white, are well represented in the auditory nerve by a peak in the response. So to every one of those increases in intensity, you see a peak in intensity. So this resembles what we saw in those step responses of auditory nerve fibers. measured by MTFs. Yes?
How come you don't show the things are aligned perfectly in time?
Oh, good question. Why don't they align perfectly in time?
Why do they align perfectly in time? I mean, don't you expect to see the correlation with the...
Oh, well, this is 3 seconds, right? So your
it's
correlation
right.
delay would be one
Right,
few
right.
thousandths of that. So it's lost on this time.
So actually there is a...
Yes, there is, absolutely. Yeah. So what the slide shows is that you sort of this transient information is already well represented in the auditory nerve, the output of the ear, and the input of the brain. This slide shows the response to another utterance with this best for making toys and gloves, and we see in the same display responses in the auditory nerve, cochlear nucleus, and inferior auditory. And so I think the response to the auditory nerve is similar in type to what we saw before. That there is some emphasis of transient, but the sustained activity is also well represented. The cochlear nucleus is broadly similar to the auditory nerve. The population is not very large, so the data are not quite so good. And I hate to say that I average all cell types. And I'm always embarrassed when I say that. But that's where things stand. And finally, in the inferior colliculus, and I would say in inferior colliculus you see a lot more transient. You see a lot more little blurs of activity brought by science, or maybe sometimes a low level of sustainability. And that's typical. So we saw two things, that on the one hand, when you look at modulation transfer functions, there's a big difference between the inferior colliculus on the one hand, where you get biphasic step responses, and the auditory nerve and cochlear nucleus, where for the most part responses that are monophasic. And here we also see in big difference in the response to speech, in that we see more transient in the inferior colliculus than at those two other stages. So maybe we can relate the two. Maybe we can use the modulation transfer function to predict the neural responses. So this is the model we used. It's actually, after doing it, we realized it was actually an old model used by Smith and Spislocky in the 70s. So it's a very simple 3-stage model. The backpass filter representing cochlear tuning, sort of sigmoid compression and rectification, which you can think of as the Hertzell response, very simple way. And finally, an MTF filter, which is derived from the measurement for each neuron. So how we're going to apply this model for each neuron? We're going to set the center frequency of the filter to the best frequency of the neuron. This stage, we sort of let it adjust a little bit to each neuron. That's one free parameter, two free parameters of the threshold and the dynamic one. And the MTF filter is determined by our measurements of modulation transfer function in that neuron. And then we put the speech and input to the model, and we get a predicted neural response at the output. Very simple. So how well does it do? Let's look first at the auditory nerve. So in the left are the measured responses, and in the right are the predicted responses in the model. And I think you'll agree that, except possibly at very low and very high frequencies where we don't have so much data, it does pretty well.
The other career data are averages, lots of averages.
Yeah, there's probably anywhere from 4 to 20 fibers per band.
So there's no difference by the time.
Actually, this may be only the high spontaneous. I forget what we did. We did it different ways. I think that is only the high spontaneous.
There's nothing systematic.
No, there are differences. Yeah, there are differences. There's not more subtle, but yes, there are. The model works for Cochlear nucleus, again, with the caveat that these are limited data and that the average on cell types. It doesn't do quite as well as the auditory nerve, but particularly in the mid-frequency range, it still does reasonably well, which is not surprising because they're not all that different from each other. And finally, in the IC. And here, well, the situation is much more grim. Well, there are some resemblances. Certainly, the predicted response is more phasic in the IC than it was in the phenomenon. So it does that right. But when you look exactly at when those pulses occurs and how long they last, if anything, the phasic character is more exaggerated compared to what you see in the actual response. So these are just qualitative impressions. We attempt to quantify things. So I correlate the model, the actual response, and the predicted neural response as a function of the best frequency of the neurons. And I do it for the auditory nerve, cochlear nucleus, and inferior nucleus. And in auditory nerve and cochlear nucleus, except at very low and very high frequencies, you get a very good correlation. On the other hand, in the IC, you get very low correlation. I would say maybe not even significant. So what this means is that the linear modulation transfer function model, as Maurice said, this is the assumption is that you can represent the envelope processing by a linear filter characterized by the modulation transfer function. It works well in the auditory nerve and cochlear nucleus, and this is consistent with what Bob Smith and Alga Müller had already said using different methods. But it doesn't work at all in inferior nucleus. So the linear model at this level doesn't work. Yes, you first, yeah.
So this is a global measure of correlation.
Yeah, it's the correlation coefficient, right? Over
The whole time.
Yeah, for the whole and
Does the IC do anything better for a particular specific systematic reading or speed?
that's a good question. I haven't.
Right, the same question, that is, if you were to look at correlations or look at the individual IC unit level, you find some units that show.
Yes, I've done that too. And some neurons show high correlations. So some neurons are more linear than others, but the average neuron is not linear. So you find maybe 20% that are fairly linear, but not all. Okay? So it says two things, right? So it says, one is that there's a major transformation on the envelope between the cochlear nucleus and the inferior nucleus. There's major processing going on, and that this processing cannot be characterized by a simple linear filter. And I did that work a few years ago, and I still haven't come up with a better non-linear model. But I think recently had some ideas, so that's still hope. But the important point, I guess, should be that there's important envelope processing in such a way that it emphasizes modulation frequencies in the 30 hertz to 100 hertz range. That's roughly. So I'm going to shift gear. How am I doing on time? Fine. I'm going to switch gear and start talking about music. So, as I just said, the IC does processing it likes modulation frequency in let's say 30 to 100 hertz. And that's interesting because that's the range of modulation frequency which produce sensation of roughness. Right? There are some sounds that sound rough, and those sounds have prominent modulation frequency in that range. And roughness is a major contributor to the dissonance of musical sounds. I mean, everything being equal, there are of course many cultural and cognitive factors in musical consonance and dissonance, but when you sort of try to eliminate those in perceptual experiment and you focus on one isolated sound by itself or musical chord by itself, whether it sounds consonant or dissonant, the main contributor is roughness. So this, so we were going to look at how this roughness is represented in the auditory nerve and inferior colliculus. So these are the stimuli that we use. So they're shown in several ways. So on the left, as so we showed the four stimuli for musical intervals. Actually, 4 pairs of musical intervals, and here they're represented in musical notation. And this column here represents in the frequency domain one variant of the stimuli in which each musical note, each of the 2 components of the interval, is a pure tone, right? So the lower frequency tone is always 440 hertz, which is what musicians use to tune their instruments. And the upper frequency is determined by the interval, and here it increases from 469 hertz for a minor second up to 660 for a perfect fifth, which corresponds to a ratio of 3 to 2. Right? And on the right, we did the same thing. Instead of using pure tones as our stimuli, we used complex tones consisting of 6 equal amplitude harmonics. So all the components are multiples of the common fundamental. The fundamental of the lower tone is always 440, and the fundamental of the higher tone, which is shown in red here or in green when they overlap, it varies exactly the same way as it does on the left. It's the same as on the left. So they have the same fundamental. They produce the same pitch between the left and right. But this one consists of pure tones, and this one consists of complex tones with 6 harmonics. And this represents results for those stimuli of this kind from a classic psychophysical study by Plomb and Levelt, where they just presented stimuli like this to listeners and asked them to rate the musical consonants of the stimuli. So the higher on the scale, the greater the consonants. And you see that for the pure tones, if you plot consonants as a function of frequency separation, you get a monotonic function. The least consonant or the most dissonant sound is the minor second, right, shown in black, which is on top here. And as you increase the frequency separation, the sounds get more and more consonant. When you use complex tone, as on the right, you get a very different pattern. You get a non-monotonic pattern. Basically, in that case, what is consonant are the intervals for which you have simple frequency ratio. The perfect fifth, which is a ratio of 3 to 2, or the perfect fourth, here, which is a ratio of 4 to 3. On the other hand, when you have more complicated frequency ratio, like the so-called tritone, which is a ratio of 45 over 32, basically square root of 2, and the minor second, which is a ratio of 16 to 15, these sounds are dissonant. So that's this observation, by the way, is what Pythagoras discovered many years ago. So we use those stimuli in experiments. So first I'm going to show data from the auditory nerve. So here we have, each column is for different stimuli, and we have data from 2 auditory nerve fibers. And the bottom are the stimulus waveform. And this tells you something, by the way, about why the stimuli are dissonant, right? This is the pure tone minor second, which we said was the more dissonant, in fact, the only dissonant of the pure tone stimuli. And you can see there's a clear beating pattern in its envelope, and this is what causes the roughness sensation, and that leads to the dissonance. For the complex tone, you get a nice waveform without any prominent frequencies in the roughness range in the envelope. So this sound is not dissonant. So the beating pattern that you see in the stimulus for the minor second is reflected in the neural response for the 2 fibers, and that you see a clear modulation of the neural activity. And this is the auditory nerve, so it encodes the file structure as well, but as I've said, this information is largely lost as you assemble the system. So for the perfect fifth, you get sort of a nice pattern without any clear modulation except for the random fluctuation. On the right, you get a response to the complex minor second. So this neuron at its best frequency near the fundamentals of the chords, which was near 440 hertz. And this one reproduces the beating pattern between the fundamentals. These neurons at a higher frequency, roughly 3 times that of the lower neurons. So its best frequency was close to the third harmonic of the complex tone. And you see that it beats also, but 3 times faster than it does here. And that's because it responds to beat between the two third harmonics of the two components of the tone diagram. But in either case, this is a dissonant stimulus and it produces fluctuations in the roughness range. So based on this, we adapted the psychophysical model of roughness, which was developed by Terhart. So what we do is, again, we produce a neurogram of the activity in response to one of the stimuli. I don't remember which one this is. Maybe it's probably the minor second, actually. So again, we have activity of all the auditory nerve arranged by frequency bands from low to high. And for each frequency band, we pass the neural response through a filter. So we pass the neural response through a filter, and the shape is a bandpass filter, which is derived from Terhart psychophysical data. So it emphasizes frequencies near 75 hertz, which is where roughness is maximal. And it's a broadly tuned filter. So this shows the output of the filter for each trace, and then we just integrate over time and take the RMS value, and then we sum for all the frequency band, and that's what we call our physiological roughness, our physiological correlative roughness. So it's the sum fluctuation in the neural response over all the frequency bands. So how well does the model predict the
Terhart filtered is from a
measurement of detection of roughness? So I ask people, do you hear it? Does it sound rough or not? So here we show the result. This physiological roughness correlate. For the pure tone stimuli, so pure tone minor second, pure fourth, pure tritone, and pure fifth. And remember that the minor second was the only dissonant stimuli for the pure tones. And indeed, it has a much greater roughness, physiological roughness, than do the other stimuli, which sound consonant. Remember that for the complex tone stimuli, the intervals, we had a different pattern where the sounds that were consonant were those which had simple frequency ratio, like the complex fourth and the complex fifth. On the other hand, the other two stimuli were dissonant because they had a complex frequency ratio. And indeed, we find the same thing in the physiological data. So in the auditory nerve data, if we filter the neural response through a filter that emphasizes those rough modulation frequencies, we are able to predict psychophysical data on musical consonance and dissonance. It's more dissonance than consonance. So after we did those experiments, it occurred to me that the roughness filter looks a lot like the modulation transfer function filter in the IC. So why don't we do that in the inferior colliculus and see maybe we will need to filter if we do it there. I think we've done 2 experiments on that. We just started this project, and this is data from one neuron. So we use essentially the same stimuli. On the top are the response to the complex tone, complex intervals, musical intervals. And we added a couple. We added unison, which has just two at the same frequency, and octave on top, which is clear what it is. And then we have minor second, pure fourth, tritone, and pure fifth. So what this shows here is, each dot represents a spike, and we presented the same stimulus 60 times, each stimulus 60 times, and we show individually as a response to 60 stimulus presentation. So here there's 60 stimulus presentation of the unison stimulus, 60 presentation of the minor second, and so on. And so there's an onset transient and an offset transient, which is kind of unusual. But if you look at that, you see that there's really sort of more activity during sustained activity, not sustained activity, more activity between the onset and the offset for the minor second, and a little bit also for the tritone. And on the right, we show two measures. One is simply the average discharge rate, the number of spikes per second, for the unison minus, for all of the 6 stimuli. And then we do see that there is this non-monotonicity for the complex tone that I talked about. The most dissonant sound, the minor second, evokes the most activity, and the pure fourth is less so, but the tritone evokes more activity than either of these. We did also the same thing with the pure tone. Here we actually used all 12 possible musical intervals to make Arnold Schoenberg happy. And again, it's the most dissonant intervals, the minor second and the major second, right, the next one, which are the most dissonant. And that's reflected both in the average rate of firing and the fluctuation in firing rate. So we got such data for about 20 neurons, and plots such as these, right, where we have the average rate of discharge and the standard deviation is basically the standard deviation of the discharge rate. So we sum them for all the neurons without selection in the IC for which we did that. And so the top is the average rate for the complex tones on the left and the pure tone intervals on the right. And the bottom are the rate fluctuation, the standard deviation. And you see again, you see the same thing for. It's particularly good in the fluctuation. It was better than the average rate. For the pure tone, we get this monotonic decrease as we increase the frequency separation. The rate fluctuation in fluctuational response diminish. While on the left, you get the non-monotonic pattern where the tritone causes more fluctuation than either the pure fourth or the pure fifth, which are consonant into it. So in the IC, we have a simpler, more direct correlate of musical consonant dissonance that we don't need a bandpass filter. It's already there. For some neurons, it's in the discharge rate. For other neurons, it's clearer in the rate fluctuation. Basically, for the sustained neurons, clearer in fluctuation. And for the onset type neurons, clearer here. Okay. So conclusion. I guess I should conclusion 1. Well, I think I spoke it. So roughness is represented in the auditory by low frequency temporal fluctuation in the discharge pattern. Bandpass filtering auditory inner fiber discharge pattern in a way inspired by psychophysics provide a metric that closely correlates with musical consonant. And then some neurons in inferior colliculus directly encode roughness in either their average rates of firing or their temporal discharge pattern, the fluctuation, without requiring a bandpass filtering. So how am I doing on time?
You're flying away, right?
Huh? Am I going too fast?
No, I was referring to
No.
actually.
Well,
I understood you have some kind of a
Yeah.
date with a taxi.
That's true, yeah. Okay.
Okay, so you got to worry
Okay,
about
fine.
yourself.
Okay. Anyway, the last I had prepared the third topic, but which is also related to modulation, the dynamic co-modulation masking release and correlate of it in the IC. But I guess with this slide we don't have time, and I will give the general conclusion, the take-home message, what I call. So I guess I tried to argue that temporal envelope properly defined, right? Temporal envelope in specific frequency bands, not the broadband temporal envelope, is important for speech and music perception. It's important for speech intelligibility, and it's important for at least consonants in music. And I'm sure lower modulation frequencies are important for hearing as you go from one note to the next. So it would be important in melody. Neuron in the auditory brainstem and midbrain process temporal envelope frequency important for speech and music. By that I mean the low modulation frequencies. And finally, so the last one is not really a conclusion, but sort of an interpretation, right? Which, I mean, we always think of speech and music as highly cognitive phenomena. Speech and music perception as highly cognitive phenomena, and of course, I will not. It's obvious that there's a huge cognitive component to speech and music, and cultural component also. And I don't want to remove anything from that, but certain aspects, certain low-level aspects, may actually reflect what goes on in the auditory periphery in the ear, as well as the early stage of central auditory processing, basically what goes on in the brainstem and the midbrain. And I'm sure that as we learn more about what goes on in the auditory cortex, thanks to our friend here, we'll find further correlates of speech and music perception in the auditory cortex as well. Okay, thank you. Yes.
So the development of, I mean, cats don't listen to speech and music, so presumably there are advantages to listening to random natural sounds in these modulations. I mean, has anybody ever looked at that? You see what I'm saying?
Which you mean, why would cats show these? So I guess the way I look at it is these are general mechanisms, right? These are general neural mechanisms which evolved long before our brains evolved to process speech and music per se, right? That's the way probably the way to think about it. So if you record it from a bat or a mouse, you would probably find or some other non-human primate, you would presumably find similar general neural mechanisms. The question is what advantage is there for a cat to have neurons very sensitive in the roughness range? I must say I don't have a good answer to that. The only one that jumps to mind is that many environmental sounds, like maybe the ruffling of leaves when someone is approaching, might have frequencies in the roughness range. I don't know. Or maybe their vocalizations have also the frequencies in that range. Yes?
Or could it be possible that the auditory system is trying to do something else, and just this perception is just like a phenomenon of trying to do something else?
That's certainly possible too, but it would be more interesting if you told me what else it's trying to do.
What about co-modulation masking?
Well, yeah, co-modulation masking is another one, but again, you're not designed to do co-modulation masking release, right? I mean, it's maybe you're designed to do auditory segregation, right? Yeah, there's another aspect of auditory perception which is very important, which bears on both speech and music, right? The ability to distinguish one's sound source among competing sound sources. And there's some evidence that modulation frequencies, low modulation frequencies, also play an important role in this ability to do so. And it's actually a very spectacular ability, right? In a busy room, we can hear one speaker, the so-called cocktail party effect, and if you go listen to opera, you can hear the soprano over the entire orchestra. So we're very good at picking one sound among others, and I suspect it's a problem that animals have to solve too, because the acoustic environment is generally cluttered. You rarely have just one sound source. So yeah, I think this whole sound segregation problem is a nice one to solve not to solve, but to study physiologically, because it is one that presumably every animal faces in its environment, from frogs to humans, right?
Bertrand,
Maybe perhaps your prediction, if you compare with optimal computer, is not so much a linear one versus non-linear issue. They rather, if it's, see, the speech signal you use to same both prediction. And the uniform material transfer function, if you clear this, the cutoff is much lower. If you limit your speech signal to that range and do prediction again, you might get a pretty good prediction.
I don't see why. No, I certainly I exaggerated when I said when I stated it was nonlinear. I guess I don't. It's possible that it's linear, but that you need to look at a wider frequency range, right? Or that you need to have one modulation transfer function in each frequency channel, and then you integrate from different frequency channels. It could be a spatial problem as well, right? A lot of those nodes have inhibitory sideband and stuff like that, and it's not clear how that would be represented. I mean, for example, is the modulation transfer function the same when you have a sound in inhibitory sideband as opposed as when you have a sound in the primary excitatory area? Probably not, right? No one has looked at that, and we have some plans to do it, but it's slow. So there's a lot of questions. I think what I can say is that a single linear modulation transfer function working on the frequency restricted single channel, single frequency channel doesn't work. That's very clear. And it's kind of obvious when you look at the neural response. I mean, you put a sinusoidal modulation and the thing fire at only one time, and you get basically one very very sharp pulsatile modulation in response. So already the response is non-linear. So it was, in retrospect, it was kind of a crazy bet to think that it would work, but maybe someone will come up with a better model. I think that's what we need to make progress.